Well, the Animal Health Committee has been extremely busy this year because of a bill that was placed in our House of Representatives concerning mRNA and identifying animals that had received mRNA injections, Representative Holly Jones. As a result, I was given a directive by the president of RCAF and by the CEO to attend a roundtable of physicians and veterinarians and PhD scientists to come back to the board of directors and educate them about mRNA. I did that. I studied mRNA myself. I wrote a paper that was, I think, Bill published it in our newsletter. Bill then condensed the paper and published it in the newsletter because we had received a scathing editorial from Drover's Journal's website that we were fearmongers and that we were the purveyors of improper information. We were gaslighted, if you know what that means. Bill responded, and they refused to publish our response because they didn't like the references. Well, the references were all identified scientific papers, and they said it would confuse their readers. I agree with that. So I'm going to give you just a little bit of breakdown on mRNA. Understand that there are no licensed mRNA injections. They want to call them vaccines. The CDC changed the definition of vaccine to incorporate the concept of an mRNA injection. There are no approved mRNA injections for any arena of livestock in the United States. The only people that are being used as a guinea pig for mRNA injections are humans. That's it. Now, mRNA is not new. It's been around a long time. It was originally established as a means of getting genetic code into a person that did not have the genetic code for a specific thing, like the production of insulin, a specific protein that would cause the body to react to a cancer protein. Now, I know that you saw in this COVID deal that all the media and all the government, everybody wanted you to say mRNA injection. Well, now you see that Moderna is advertising an mRNA on TV about every time I turn the thing on, and there's a little gal climbing up the stairs, and at the top, mRNA for cancer therapy, and all of a sudden again, mRNA is gene therapy. So it can be whatever they want it to be, I guess. I want to give you just a little rundown. There are mRNA injections being given to poultry and swine in the United States under a provisional license. What is a provisional license? Well, a veterinarian may go into a swine facility and identify a virus that's causing trouble there. Maybe it's circovirus, maybe it's influenza A. They then take that virus and get permission from the USDA under the prescription of a licensed veterinarian from that particular state and manufacture the mRNA. The interesting thing about this technology, although it is quite expensive, you can multiply the dosage on these things very rapidly, and you can form an mRNA to give as an injection in about 8 to 12 weeks of the time that you put the virus in the system. In other words, if you've ever had a Bactrin made up on your own farm for a specific bacterial condition, such say pink eye or salmonella, we call those autogenous Bactrins. Autogenous is Latin for auto-generated. It means it comes from your farm, and it can only be used on your farm for that specific problem. And now we have autogenous viral mRNA injections. That is going into the swine industry and going into the poultry industry. And that is why Representative Holly Jones presented to the State House of Representatives a bill to require that if anyone was doing that, that the meat be identified. So why was she interested in identifying the meat? Well, in the processing of mRNA, they have changed one of the nucleic acids on mRNA to it's called a pseudouracil. I'm not going to get into all the biochemical dynamics of this, but uracil is one of the 4 nucleotides that RNA uses to gather the code from DNA. And most RNA doesn't stay around very long. If it did, it'd keep manufacturing the same thing over and over and over and over. Well, it has a system by where it's decomposed. There are enzymes designed to break it down. Well, by putting pseudouracil in, you stop that from happening. In other words, an mRNA strand that might last a few hours or maybe a day or two now lasts 6 months, 4 months, 3 months. In some cases, they've documented up to 8 months. The problem with this pseudouracil in an mRNA injection is it causes the body to keep manufacturing what is coded on the mRNA. I don't want to get too deep in the weeds, but in other words, the pseudouracil doesn't allow it to be broken down. And so when Representative Jones found this information out, she was concerned about people consuming this mRNA and then the mRNA having function within the human body to continue to make this viral pathogen for which it was coded. I'm going to go to just one slide and demonstrate a little bit here. I'm not sure how to run this. Press the big one, I bet. All right, this is the only slide that I'm going to show you today to give you a little bit of an understanding of what mRNA does. You know that your genetic code is unique to you. Every individual genetic code is unique. That's quite interesting. You can have the exact same genetic code in a pair of identical twins. One egg has been fertilized and it's split in 2, and 2 individuals have developed from that specific egg. Their genetic code is identical, and yet they are completely different. They may have a different personality. One may have a mole here, another one a mole there. We found out in cattle when we cloned them, we'd have some with a star in their head, some with a blaze. You know what that's all about? It's called genetic expression. We only use about 10% of our genetic code at any one time, and often we only use 10 or 15% through our entire lifetime. And DNA contains the genetic code for making proteins. That seems quite unusual. The other thing that's unusual is the genetic code in the wood that makes up this podium is made up of the same type of material as what's in you or your dog or your rabbit or the deer you kill or the antelope that you shoot on your property. A common creator, a common system. Well, in that DNA, there are nucleotides. I'm not going to get too into the weeds here, but mRNA is a compound that goes into the DNA and gathers up the code. And every 3 nucleotides codes for an amino acid. I'm sure you've had animal science. You know there's 20, right? Essential amino acids, and there are some branched amino acids. Well, these amino acids are united together biochemically to produce a protein. And then these proteins are folded up very, very significantly. The best I know to explain that is the inside of a baseball is a good example. If you're like me and you was a kid, you might have knocked the cover off a baseball. You used it too hard, or you kept throwing it up on the barn. It finally got hit by the tin and cut, and it come apart. It's all wound up inside, strings on strings. Well, that's what a protein is. So mRNA basically codes for a protein or an enzyme, which is also a protein. And it goes in, gathers the code, comes out to the ribosome. The ribosomal RNA reads the mRNA, transfer RNA brings the amino acids. They form the protein. It gets folded up. And so what our COVID injection did is they had mRNA that was coded for the spike protein that was on the outside of the coronavirus. If you look at coronavirus under a high, like electron microscopy, you can see these things. And they're just protein projections that allow the virus a better chance of getting attached to a membrane. And so you had mRNA that went in you that coded for that protein. It was given to you as an injection. It went into your muscle cells, your lymphatic system, And as it was absorbed into those cells, it began to program those cells to make this spike protein. And it made the spike protein over and over and over, and your body says, that don't look right. And so it makes an antibody to this spike protein and supposedly immunizes you and does a pretty darn poor job of it. Part of the problem with long COVID now has been identified that you might have taken one injection, not had any problem, but then you take the next one, and then you take the next one, and then now they're still wanting you to take them at home. Public Health Department sends out a thing on the radio every morning when I listen to the news to listen to the funeral announcements. Come and get your COVID booster. We recommend them every 90 days, every 6 months. I guess they want you to be boosted from now on to eternity. But when that happens, that pseudo-uracil stays around and keeps coding for the same thing over and over. And the spike protein is toxic. Your body doesn't like it. It rebels against it. So mRNA, you can grow this mRNA by infecting bacteria with this virus. The bacteria then make the mRNA, then isolate the mRNA, and you have an mRNA injection. So far, we don't have the ability to synthesize any kind of nucleic acid. You can't make DNA, you can't make RNA. And so that's what an mRNA injection does. We do the same thing with any species. We identify a protein antigen, we get the code for it from mRNA, and most of this mRNA is just viral pathogens. We don't need it really for bacteria because they got DNA. But a lot of RNA viruses, and there are DNA viruses that we can get the protein antigen off the capsule or something like that. And so we have injectable mRNA. One of the issues with mRNA is it doesn't stay in the body. It is quite well documented now that the mRNA is expressed out of the body, even from people that have been injected to people that have not been injected, similar to how you would transfer a virus to one another, sitting at a table, holding hands, kissing, any kind of close association with other people, you transfer it. That's how COVID basically infected all the people it did in the United States. Well, the mRNA does that in and of itself because the body tries to excrete it through extracellular vesicles. These are mRNA pieces that are surrounded by proteins that the body then wants to expel. It can be expelled in sputum, it can be expelled out of your nasal cavity and saliva. There's even some indication that it can be transferred venereally. So I could go on and on. I don't know near enough about it. It is a not unique technology. It's been around a long time. I would suggest and give you an idea that get on the internet and look for Dr. Malone or Dr. McCulloch. They are two MDs, well documented, did a lot of research with mRNA when it first came about 30 years ago. Each one of them puts out a little blurb every morning associated something with mRNA. They are physicians, they're not veterinarians, but you can get the headline and the first 3 or 4 paragraphs of what they're publishing free of cost every day, or you can join their web page. I think it's $5 a month, and you can get all the scientific data you would ever want to receive in a lifetime in about a week. Very well-respected scientists and physicians that are providing information to the public that the government and other sources are basically not releasing. Any questions? Malone, M-A-L-H-O-N-E. McCulloch, M-C-C-U-L-L-O-G-H. You know, on such an important subject, you thought they'd give me a little more time, wouldn't you? Thank you all very much.